Have you registered now? Have you registered yourself in the class?
Yeah.
Yes. Okay. Because you missed the first class, so very briefly then I will tell you, in case you're a little lost, this is basically of the human body. So basically we will touch on anatomy and physiology. A little more on physiology than anatomy. Okay. But even though we are studying a little of anatomy, we're not dissecting any animal. We're not opening any animal. We're not opening any dead body either. So we are concentrating a little bit more on physiology, how the body works. Please, can you do me a favor and close the back door? Maybe I don't think the other students may be coming. Can you briefly then tell me what is your major?
Human health sciences.
Okay. Are you studying to be a nurse, a doctor?
Health check. 臨床検査技師.
Okay, but that is not a medical doctor.
Not the real doctor.
Mm-hmm. Okay, so I didn't bring the survey. Usually in the first class I ask the students why they are taking the class. Especially to be clear a little bit on what they expect and what are we going to do in class. Anybody else who is the first time?
First time.
Oh, okay, so the three of you. Can you briefly tell me then, sorry, your first year grade?
Yeah, first year.
Okay.
Second grade.
Second, okay. What is your name very quickly, your name and your background?
Name? Kaito.
Mm-hmm. And background, what is your major?
Pharmacy.
Pharmacy. Okay, so you both are health related. What about you? Yes. Very quickly only your name and what year grade you are.
Human health science and physical therapy.
Oh, okay. Is this your first year? First year. Okay, thank you very much. Well, I don't know if you will end up registering or not, since this is your first class. We already covered very briefly about the history of anatomy and physiology. Okay, not really into the main content of the course. Just to be clear that we are not going to do any dissections, so not opening any animals, not opening any body. Unfortunately, I may not be able to bring also some 3D models perhaps, so we will have to look into, you'll have to look into some books if you want to check more slides, more figures, different perspectives of the systems of the organs, okay? There's a lot out there on the internet. You can watch also videos and there are many books. Last class I recommended, well not recommended, but I mentioned a book that I'm going to base the classes on. It's Anatomy and Physiology. Okay, but you can look any anatomy and physiology book. I suggest you look for these books that are anatomy and physiology because they are not too heavy on anatomy or too much on physiology. The idea for you is to be exposed to English classes, maybe to hear English terms, medical terms. Perhaps many of what you're going to study this semester here, because you're from medical science, probably you know them already or you have studied them in Japanese. So the idea is not to make the course too difficult, but you will have to speak a little bit, write a little bit. Part of the grade is also to present and to write a very short essay. We have a final exam, but it's supposed to be based on the classes, the handouts that I give you every class, and it's a lot of memorizing. Okay, so if you find new terms, new words, try to practice writing them out. I'm not qualifying your English ability, so as long as it's understandable, it's okay. Okay? So don't worry too much if it's correct verb tense, the perfect sentence, and so on. Just try to do your best. It's better always to maybe participate if we have discussions or group works, and so on. Anybody who couldn't swipe the ID? No? Everybody swiped. Okay, thank you very much. So I don't know if the person who mentioned one of their expectations last class is here or not, but just for the sake of information for all of you, what we will not cover in the class is about nutrition. Okay, so nutrition is really not related to anatomy or physiology. But I know there is another course about nutrition, so you can check up the syllabus if you want to know about nutrition and take that course. And also we are now going to learn how to respond when we are injured or we are sick. So if you wanted to have some knowledge about prevention maybe, or first aid or something like that, this is not that course. Okay, so very quickly then, can you hear me on the back? Is it okay? Can you hear me? Yes? If you want me to repeat more what I say, let me know, okay? I don't want to keep talking, and then nobody really is following. So if you don't follow, please let me know. Please talk a little slower, repeat the questions, repeat the slide is okay. I try to put a lot on the slide so that you can read and then follow what I'm saying, okay? So sorry, I am with allergies, so I guess some of you are also with allergies. Maybe my voice is a little bit funny. So very quickly, what is important about knowing the definition of anatomy? What is anatomy and what is physiology? Are they different? We briefly touched on this last class. Anatomy is the study of structure and shape. Okay, so that's the basic idea. Anatomy is the structure and shape of the body and its parts and the relationship to one another. But physiology, when we mention physiology, is how the body and each of its parts work or they function. So they are different. Okay, one is the structure, the shape, the other one is the function. Now if we stop for a minute to think about how many millions and millions of cells are in your body, they're constantly having activity. All your cells in your body are having some activity. And there is most of the time we are healthy, everything is going fine. Little is actually going wrong. So all the cells in your body have to work a lot really to maintain your body within some normal range, right? And this is amazing. This is something called homeostasis. Okay, have you heard this word before?
Yeah.
Yes? What about the other students? Have you heard the word homeostasis? So homeostasis means the body's ability to maintain, to keep relatively stable internal conditions despite large changes in the external environment. So imagine that external environment changes a lot, but inside your body or inside each of the cells, they have to keep things working within a normal range, right? Where does the word come from? We mentioned that most of the medical terms come from Greek or Latin. Homeo means the same or unchanging, and stasis means standing still. But does the term mean that that things are unchanged, that it's an unchanging state? What do you think? Any thoughts? Is it an unchanging state? Those who say yes, please raise your hand. And those who say it's not an unchanging state, raise your hand. Oh, yes and no.
External state. For example, it's very hot.
Exactly.
You sweat,
Yes.
and temperature is unchanging.
Okay, it's actually doesn't mean that the unchanging state. It's very dynamic. It's a dynamic state of equilibrium where you're trying to have is your body to be within normal range. So despite or even though there are many changes, it's a balance in which the internal conditions, it's a balance in which the internal conditions change and vary, but within a narrow limit. Okay, and this is the important part. It's within narrow limits. If we try to make a graphic about these, homeostasis, we reach or we can achieve homeostasis using mainly the nervous system, the electrical signals of the nervous system, and the hormones from the endocrine system. Okay, but the stimulus is the change in the environment, and despite the type of stimulus, all the homeostatic mechanism has basically these three elements: the receptor, the control center, and the effector. This is to simplify all homeostatic mechanisms. They will at least have these three components. Okay, so what do you think the receptor will be in charge of? Anyone?
Sensation.
Yes, that is also part of it. They will sense the change. They will sense the stimulus. Okay? The receptor will sensor and will monitor and will respond to the change in the environment. The change in the environment is the stimulus. Okay? So we receive a stimulus, the receptor captures this, notice this, and what will the receptor do? Will send the information to the control center. Okay? This is the basic idea. The receptor finds the stimulus, sends information to the control center, and these are very specific terms. All the information going from the receptor to the control center are called afferent pathway. Okay? Everything coming in, the input, is going to be afferent pathway. And we will see a little bit of this also when we study the nervous system. They also call it afferent pathway. So the second component is the control center. The control center will determine or will decide what to do. Will analyze the information and will decide on the appropriate response. Makes sense, right? And then it will send an output information to the effector. Effector is actually who will do what through afferent pathways. So all the pathways going from receptors to control centers are afferent. The ones going out, carrying the output information, are called efferent pathways. Okay, so the output information is sent through these pathways to the effector, and the effector will conduct a response. But the result of this response will influence the stimulus, will give you feedback You have heard probably the word feedback, right? When we ask you to tell us about something, to give us comments, for example, we say, please give me feedback about something. So receptor monitors, notice there is some change in the environment, sends the information to the control center. The control center is okay, says, let's do this, sends the information to the effector. The effector conducts a response, and in turn we receive some feedback. Depending on what kind of feedback we receive, we may call it positive or negative feedback. Have you heard about positive or negative feedback before?
Yeah.
Anybody else? No? Is this the first time? Okay, so lucky for us, most of the processes in the body are usually negative feedback. Okay? And why are they called negative or positive? Would you like to share? In your own words. It's related to the stimulus, right? We receive a lot of stimulus. We have this mechanism. Take your time, it's okay.
Express the control center's function.
Yes. So the negative feedback will influence actually the stimulus to depress it. That is why it's called negative. The control center will shut down, the stimulus will decrease. That is why most of our mechanisms are usually negative feedback. We will see some examples to make this a little more clearer. Negative feedback will change the direction of the stimulus. Okay? Either increase or decrease, but will change the direction. We mentioned most of the common mechanisms in the body are using negative feedback. Heart rate, blood pressure, breath rate, or levels in the blood of glucose, oxygen. For example, if you are, after lunch, you ate a big meal, what happens in your blood? Your glucose level will rise, right? Glucose will go up because you ate. So these will trigger the pancreas, one of our organs, to release insulin, okay? Because it's not good that our blood has high levels of glucose. These can damage organs, so it's not healthy. So when we eat, we raise the levels of glucose. Our pancreas says, okay, I will release insulin. Insulin will make the liver to process the glucose. Okay, so what do I do with the glucose? Okay, I will store it, I will keep it. For times when I need glucose, I will keep it as glycogen. But if I keep it as glycogen, that means that less glucose will be available in the blood, so decreasing the levels of glucose. Correct? So a rise in the glucose level causes now a decrease in the glucose level to maintain everything within normal range. And the opposite happens the same. If we don't eat, if we're starving, if we skip a meal, 2 meals, 3 meals or more, that means we're fasting, right? We're not eating. So the levels of glucose will be diminished. They will go down. And that is also not good, right? You will maybe start feeling dizzy, your organ will not have energy enough to function, and so on. So what does our body do within certain limits? But what our body do is to release again, the pancreas will release another substance called glucagon. And what you had stored in your liver, okay, says now it's time to release this glucose that we have been storing as glycogen to raise the levels of glucose in blood. Okay, so the body tries to keep glucose levels within a range, a normal range. Okay. This is with figures. Can you still hear me from the back, even if I put the mask? Yes, I assume. Okay, so you can check on these figures that I took. Most of the figures that I will show you are taken from this book, from Pearson Education. Okay. For those of you who are coming for the first time, if you are going to stay in the class, I usually keep extra handouts. So you can come to my office, I will give you extra handouts. Okay? Unless you want to photocopy from some of the other students. So as we mentioned, homeostasis, the purpose is to keep the glucose level within normal range. Okay. What happens if we have an imbalance? Whenever we eat, we will raise the level of glucose. That will stimulate the pancreas, certain cells, to release insulin. The main thing will be happening in the liver. The liver will take up this extra glucose and will store it as glycogen. All of our cells in the body will also start taking up glucose, will start using the glucose. And the blood glucose will decline. Okay? Will decline to a point where the insulin will start diminishing. So it's no more necessary to keep releasing insulin because the body is returning to the normal state. Right? The opposite will happen when we fast or when we skip meals or we don't eat. As I mentioned, there are a few examples of positive feedback. It's rarely happening in the body because basically here there will be an increase in the stimulus. So something that triggers all these control will increase. So it's not very good for the body. Basically, it will maintain the direction of the stimulus, okay, and can even accelerate the effects. We mentioned it's an infrequent event, and these are very two clear examples. We will see, imagine childbirth. We all know a little about childbirth, okay, when babies are born, when the mothers are delivering. This is an example of a positive feedback, the uterine contractions during childbirth. Uterine is this organ where the baby is growing inside women, okay, pregnant women. So when it's time, and you have seen many in the movies or maybe from some experience, the women is starting to have contractions. They start to have these horrible pains, very, very strong pains, because the uterine is starting to contract. And it has to contract because it's time for the baby to come to the world, right? So we need to expel the baby from there. But some hormones, oxytocin, is causing the uterus to contract. So if we have increasing levels of oxytocin that is contracting the uterus, and it's terribly painful, do we want to shut it down or no? If we would have a negative feedback, would mean that we will decrease the level of oxytocin, but in that case the baby will not be born, right? In that case, the baby will die inside and we will need to do C-sections to all pregnant women. But what happens? Oxytocin causes the uterus to contract, the baby will start coming down, and while it's stretching the cervix, this is the cervix, okay? So the baby pushes, and this part is stretching. And this stretch causes some nerve impulses, sent to the brain, and the brain will stimulate another organ to release more oxytocin. So this is a positive feedback. Okay, so we have an increase in oxytocin and we are stimulating, and the end result is more oxytocin. Why? Because we need all this to happen. So you see there is very different negative feedback from positive feedback. Okay? So every time you think about negative feedback and positive feedback, you can maybe think of the childbirth and when you eat or you don't eat. That may be more simple examples. So much of the following, probably you have studied at school. Did you take biology class? Did you study biology in the Japanese school system? Yes?
Only basic class.
Basic, okay? Don't worry, this is also basic. So it will be a review of what you probably already know. We know already that cells are the smallest living unit, right? They are made of mainly these 4 elements: carbon, oxygen, hydrogen, nitrogen. And what is amazing is that the living cells are mainly made of water. 60% of them is composed of water. So water is very, very important, and we all know this, to survive, to live. And the major building material of these cells is the protein. Okay? So protein is the main component of cells. We very briefly mentioned the cell theory last class. These are the 4 more important things that you should remember about the cell theory. Probably you already know these from your previous classes. The cell is the basic structural and functional unit of all living organisms we mentioned. Okay? The activity of an organism will depend on the collective activities of its cells. It makes sense, right? It's not one individual tiny cell doing something completely different from the next cell. Biochemical activities of the cells are dictated, they are decided by their shape or form and by the relative number of their specific subcellular structures. Today we will go very quickly on some of these structures and the shape and form. And of course, continuity of life, so to continue life, it has a cellular basis, right? It's one cell producing more cells. So think about now, try to remember going back to your biology class, and what are some parts of the cell that you remember? To make this less difficult and lonely, let's try to do it in groups. We have maybe 4 and 4 people. If we can do 2 groups. Unless you want to do 2 and 2. 3 minutes. Can you do 2 groups? And think about some parts of the cell. Yes? Maybe you can sit here if you want. You're already sitting very near to 5 here and the 3 of you here. So you don't move too much. We are sleepy after lunch. I want to see how much you remember, okay? Because this is the only class we are going to touch on this. Every class is only that class, so let's see how much you remember. Parts of the cell. Let's go. See, you will have to talk to each other. And you have 5 minutes. Do not cheat. Do not cheat. It's just an exercise. If you don't remember anything, then you say, I don't remember anything. I was sleeping through the whole biology class.
Oh yeah.
Nucleus whole. Is it part of the cell? Nucleus has chromosomes. Oh yeah, it's part of the cell.
Okay, so that's your limit. Yes?
Yeah.
Okay, then let's share with everybody how much you remember from parts of the cell.
9.
No, that's good. Okay, what kind of things you remember about the cell?
List or
Yes, just tell me what you remember.
nucleus,
Okay, yes.
cytoplasm,
Uh-huh.
endoplasmic reticulum, cell membrane, ribosome, lysosome, mitochondria, Golgi apparatus, chromosome.
Amazing. Were you looking the next slides? No.
Next.
No. Okay, that's good. Do you remember anything else?
Four.
Okay, that's good.
Cytoplasm,
Mm-hmm.
cell membrane, mitochondria.
Okay, good. So at least we remember some words, some things about the cell. It's okay, you can go back to your seats. This is a little bit only just like an exercise to see how much you remember. For you to speak also. Otherwise, you are sitting there and will not talk to anybody else here. So you need to speak a little bit. And then write down, because both groups made the list, so that's very good. Thank you very much. So let's see a little about. There are many types. We mentioned the cells are the basic parts, the unit, or the living unit. But there are some common things to all the cells. Cells have the same basic parts. We mentioned many of them. And there are certain common functions to all these cells. Okay? We mentioned these three main regions, or three main parts of the cell, if you remember. We mentioned the nucleus, right? This is the place where all the activity takes place, reproduction. It's very important because it has the map or the blueprint to build the whole living organism. So without the nucleus, we cannot build another cell, yet alone another organism. Right? So it's very important, the nucleus. The cytoplasm, with the cytosol, the organelles, and the inclusions. We will see very briefly about each of them. And of course, the plasma membrane, right? The plasma membrane will delimit, will enclose the cytoplasm. Otherwise, everything just go swimming to another direction. So we need to enclose everything here. It's like having this room, and the wall would be the plasma membranes or the cell membranes. We have the control panel here to reproduce the same room, would be the nucleus. So these are the very basic things. Now pay attention. I hope the next slide is a video. So pay attention to the video. I hope it runs smoothly. If it's too fast, at the end you can find the links of the videos that I'm playing today. They're available on YouTube, so you can listen to them again and again and again if you want. Okay? Let's go.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, And smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material.
For you, this is the most important.
And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
You can forget it. Okay. So this is to give you a more visual idea of the parts of the cell, some of the organs here, to make it less boring. Not all of the videos, the videos are not perfectly fit, maybe all the information for the class, because I'm taking them from the YouTube, but I'm trying to take the ones we have the most appropriate or relevant information for us, okay? So about the plants and so on, well, you don't need to know that. It's just for background. So we mentioned about the nucleus, and we mentioned how it's the control center. Something important also mentioned in the video, and is that it will include the genetic material. Okay, maybe you have read before DNA. Have you ever wondered what it stands for? DNA, deoxyribonucleic acid. Okay, most commonly called DNA. From now on, we will only say DNA. And the nucleus has these three parts or regions. The envelope or the membrane, okay, the nuclear membrane, which is a double membrane. It's similar to the cell membrane, but it's double, meaning it's twice. That allows some, but not all, the substances to pass through, meaning to pass through this nuclear membrane to go from the cytoplasm inside the nucleus. Not all the substances will pass through it. It encloses the nucleoplasm, in which other elements are suspended or are like floating or swimming. Okay. The nucleoli, the little nuclei, one or more rounded bodies where ribosomes are assembled or they are built. Okay. The chromatin we mentioned, it was mentioned also in the video, is this network. When it's loose, we call it chromatin. It's the threads of DNAs with proteins. Okay, so it's loose when it's not dividing. But once the cell has to prepare to divide, then it starts condensing and it forms the chromosomes. The cytoplasm is the place where most of the cellular activity will take place. It has these three major elements: the cytosol, the organelles, and the inclusions. The cytosol is the fluid that suspends the elements. So it's the fluid that contains the elements. Okay, mostly it's made of water. The organelles, such as, and you mentioned many of them, mitochondria, ribosomes, endoplasmic reticulum, lysosomes, and so on. And the inclusions, some chemical substances that may or may not be in certain cells. Most are stored nutrients or cell products. This is just a, yes?
I didn't understand the inclusions.
Inclusions are some substances that sometimes are contained in the cells.
Substances?
Yes, like granules, glycogen granules in the liver or melanin in the skin. They're called inclusions. Maybe we will see a little of them while we study some of the cells. We will study certain types of tissues and with that the cells. You mentioned the plasma membrane. Of course, plasma membrane is way more complicated than only one slide. I'm trying to oversimplify the information for you. All of you have a basic idea of what the plasma membrane is, right? It will contain the content of the cell. Okay, so it will surround the cell, the nucleus and its contents, the organelles and all other elements. It will separate the cell from the environment, from the surrounding environment. Okay, and it plays a dynamic role in many activities. So it's actually not just like walls without a function. And it consists of two lipid layers, has two layers. They are arranged tail to tail, in which the proteins may float. So it's why it's called a fluid model. You see here, it's these layers here are the heads and the tails, heads and the tails, if you remember from your classes maybe. Why they are arranged in this way? Because the heads are hydrophilic. Remember on the outer, it's surrounded by water. So the heads are hydrophilic. but the tails, the inside, actually does not like water. Okay, they're hydrophobic, meaning hydro means water. Okay. So like this picture, some proteins may be scattered, maybe in different places of the plasma membrane. And they are responsible of different functions. Some proteins may act as enzymes, others could be receptor or could be channels so that substances can go through the plasma membrane, and so on. But how does one cell will attach to another cell? Otherwise it will just go floating. There are very few cells that will go around moving freely, like the blood cells, for example. Most of the cells have to be bound together, and they may be bounded in 3 ways. It's called a gap junction, desmosomes, or tight junctions. In the gap junctions, the neighboring cells will be connected by these cylinders of proteins that will go from the entire width of the membrane. They span the entire width of the membrane. Desmosomes, they are these filaments attached to a structure that is like a button. It's a button-like structure. And they are connected through these filaments to the neighboring cells. And we also have a third type of junction called tight junction. These are different types of junctions. They are impermeable. Do you know what means impermeable? Yeah.
It cannot be entered, right?
Mm-hmm.
Yeah.
Yes, that's right. It will refrain substances from going from one side to the other. So they are fused together, the cells are fused together, and they will prevent the substances from passing. Imagine the digestive enzymes in the intestines. If we wouldn't have these kind of junctions, they will go through and go to your bloodstream, to your blood, and that would be very dangerous. Okay, so depending on, again, the function of the cells, we will have different types of junctions.
The plasma.
Okay, sorry. Again, next one is a video. Please listen carefully. Again, if there is something that is not clear, the link is at the end of the handouts. You can re-listen again, watch again the videos. Okay?
The plasma membrane is made up of several organic molecules. I will concentrate on 2 of them in this presentation. The first molecule is a lipid. It is called a phospholipid because instead of the usual 3 fatty acid chains, there are only 2. The third bonding position is with a phosphate group. The phospholipid molecule is divided into a head region and a tail region. The head region is the glycerol and phosphate group. The tail region is the fatty acid chains. The head is hydrophilic, attracted to water. The tail is hydrophobic, repelled by water. It is this property which allows for the unique molecular alignment that makes the plasma membrane. If a droplet of phospholipid was put into water, a mycele would form. A mycele is a sphere with the hydrophilic heads on the surface, the tails inside, away from the water. Our bodies are made up of approximately 60% water and trillions of cells. The water can be found inside the cells, outside the cells, and in the bloodstream. The location of water and the phospholipid, hydrophilic, and hydrophobic ends enable the membrane to form a bilayer of lipids. The currently accepted model of the cell membrane was developed by Singer and Nicholson in 1972 at the University of California, San Diego. Pictured here, the plasma membrane looks like a sea of lipid with icebergs floating in it. The membrane is very flexible and has a fluid motion. This design of the plasma membrane is called the fluid mosaic model. Quite often, these proteins have sugar molecules attached to them, forming glycoproteins. These molecules extend from the membrane surface and act as an identification marker. The proteins are used by the immune system to identify a cell as being self, or foreign called non-self. Each individual has their own biochemical makeup. In other words, each human being has their own set of cellular proteins. But if a cell comes into the body that has a different set of proteins, whether it is a bacteria or an organ transplant, the immune system may identify it as self or as non-self and attack. In order for the cell to survive, many different materials must be able to cross the plasma membrane. When water passes through a cell's membrane, the type of diffusion is called osmosis. When solid particles pass through a cell's membrane, the type of diffusion is called dialysis. Before looking at this movement through the membrane, it is important to understand the movement of molecules in general. Atoms and molecules are in constant motion. This random motion will disrupt the cluster rather than maintain it. This motion is referred to as Brownian movement. This motion is always present in some degree, in gases, in liquids, and even in solids. For instance, if a bottle of perfume is left open in a room, eventually, no matter where you are in that room, you are able to smell the perfume. The molecules of air hit the liquid, causing the liquid molecules to move into the air. At the time the bottle is first opened, there is a high concentration of perfume molecules in the bottle, a very low concentration of perfume in the air. As the perfume molecules are bombarded by the air, the concentration of perfume is dispersed into the air. In other words, the perfume is moving down a concentration gradient, high to low. This movement is called diffusion. It uses Brownian movement. Now let's take a look at another example of diffusion. In this aquarium of water, we drop some blue dye. As the water molecules move, they bump into the dye molecules. The dye molecules, at first in a high concentration, start moving and bumping into each other and the water molecules, moving into the area of lower concentration. As this movement continues, the dye spreads out and equilibrium is attained. Equilibrium is equal concentrations of the dye and water in every area of the aquarium. Remember, Brownian movement continues even after equilibrium is reached. Although extremely thin, the plasma membrane separates the contents of the cell from the environment. The membrane has several functions. This video concentrated on the movement of materials through the plasma membrane. Remember that this membrane is selectively permeable and can prevent materials from passing through or keeping materials in, and it will permit, even facilitate, the entrance and exit of materials. Materials may move through membranes passively or actively. In this video segment, passive transport, which uses Brownian movement, was introduced. Passive transport includes diffusion, defined as the movement of materials from a region of high concentration to low concentration. Two specific forms of diffusion are osmosis and dialysis. These processes are essential to the survival of the cell. Nutrients must enter, waste must exit. The structure of the membrane allows for the processes to take place, which enables the cell to live.
Okay. So you heard in the video selective diffusion, selective movement. Did you catch that one? So that means that some molecules may pass and others don't. Okay, that is selective. So you are selecting which ones will pass, others will not. Okay, so you mentioned many of these organelles. You mentioned the mitochondria, ribosomes, the reticulum, Golgi, lysosomes. So I'm glad you remember maybe most of them. We will go over the general idea of them, what's the main function, okay? Because that will explain why some cells may have more of these organelles and less of others, okay? Each of them will have a specific function, so let's concentrate on that. You may have seen many pictures of mitochondria, right? You remember from your classes? This is a mitochondria. It's a wall, it's double membrane. By double membrane means here is the membrane and here is the second one. Okay? That are placed side by side. Okay, the outer membrane is smooth, but the inner membrane has protrusions. Protrusions are these accommodations, look like these, and these are called cristae. Okay, and this is where the ATP or adenosine triphosphate will be produced or synthesized, will be produced. The ATP is like the money or the energy. This is the energy that all cells need in order to be able to carry out their activities. Okay? So that's why many times the mitochondria is called the powerhouse of the cell, because they will produce all this energy. Certain cells will have more mitochondria because they will need more energy in order to carry out their functions. One of those cells is, for example, the liver, where you have to, for example, is in charge of processing drugs or the food, glucagon, and so on. Muscle cells, they need a lot of energy in order to contract and produce the movement of the body. So liver and muscle cells have a lot of mitochondria. We mentioned this. ATP stands for adenosine triphosphates, and ATP will provide a form of the energy, the chemical energy that all the body cells will use, okay, to carry out their functions. The ribosomes are these tiny, do you understand the term bilobed? Has two lobes, yes. Mm-hmm. You want to say something? Yes.
Lobe is
Exactly.
that.
Mm-hmm. So bi would mean having two of them. Okay. They're made of proteins and one variety of RNA called the ribosomal RNA. These are the actual sites of the synthesis or the production of the proteins in the cell. So they are very important for the cell. Remember, the cells contain a lot of protein. Okay. Some of these ribosomes may be floating freely in the cytoplasm, and others will be attached to membranes, for example, in the rough endoplasmic reticulum. What is the endoplasmic reticulum? Do you remember? At least a picture. Yeah? This is like a system of cisterns. They are filled with fluid, and they are coiled and twisted. They are twisted through the cytoplasm. It's actually a network. It's like a whole channels network to carry substances. You will move substances from one place to another place in the cell. Okay? We may have also the rough endoplasmic reticulum. We mentioned they will be, why it's rough, they are covered with the ribosomes. They are the cell membrane factory, and they are abundant, which means there's a lot of them in cells that make and export proteins. Okay? On the other hand, if they don't have the ribosomes attached, then they're the smooth endoplasmic reticulum. But these smooth endoplasmic reticulum has no role, they have no part, they do not involve in the synthesis of protein. They're involved in the lipid metabolism, okay, and detoxification of drugs and some pesticides. So they're abundant in liver cells. You can, with all the time, look into the pictures, and I would suggest that maybe you go into some textbooks if you want to see more pictures. Textbooks, or just try to maybe look for images so that you have a better idea Okay? The Golgi apparatus, there are sacs, membranous sacs, they are stacked, they are one over another, and they are associated with groups of very small vesicles. Okay? This is a Golgi apparatus, if you see this here. They are very close to the nucleus. And the major function is to modify and package proteins. So they will move the proteins also from one place to another. The lysosomes are some membranous bags containing some enzymes, particularly digestive enzymes. So they're able to digest worn out or old or non-usable structures from the cell and foreign substances. Foreign substances mean substances that are not from the cell. Okay? They are particularly abundant, numerous. You can find a lot of them in certain cells called phagocytes. Okay? The peroxisomes are similar to lysosomes, but they have different enzymes. They use molecular oxygen to detoxify some harmful substances, such as the alcohol. The most important function of peroxisomes is to process free radicals, which are harmful to the cell and to the body. They are numerous. They are abundant in the liver and kidney cells, which are very active in detoxifying processing. They look like very small lysosomes, but they do not detach or they do not arise or originate from the Golgi apparatus. Instead, most appear to come from the endoplasmic reticulum. Okay? This is just very general information of all of them. The cytoskeleton we saw a little bit on the video. This is a network of protein structures that will go throughout the cytoplasm. They are sparse in all the cytoplasm. They are like the bones and the muscles of the cell. Okay? If they are the bones and the muscles, then they help determine the shape of the cell. All the cells may have different shapes, and we will see a little of the shape later. The cytoskeleton will determine the cell shape. They support also the organelles, and they provide the intracellular transport and help with some cellular movements. They are made up of microtubules, filaments, and microfilaments. Basically, all of them made of protein. Very quickly, we saw in the video also there are some cell extensions, like the cilia that we can see in the digestive system. They will move some substances throughout all the lining. Microvilli. These are tiny, what is finger-like extensions? We will see many times something like this word. It means only like fingers. Okay? So these are extensions or protrusions that they look like fingers of the plasma membrane. They are projected from an exposed cell surface. Well, we have seen some images of the cell, but actually they come in different shapes and forms, and they have many different functions. But the shape and the relative number, we mentioned these, of the organelles that are contained in a cell are related to the specific functions of this cell. Okay? We have different types of cells, like blood cells that we call erythrocytes, epithelial cells, the sperm cell, the nerve cell. This is only one cell. muscular cells, and so on. So there are different types, different shapes with different functions. And here is where we are going to see a little of how they work. Okay, so now we have reviewed the basic of all of them. They all have these parts, they all have these elements. The elements function in this way. Now we will see a little how they work together. So most cells have the ability to reproduce. They will divide, multiply, so on. They will grow. They will move, metabolize, which means process. They will digest foods, excrete wastes. What means excrete?
Expel. Expel.
Another?
Get rid of.
Exactly. To get rid of the waste. Okay? And respond to stimulus, which is called irritability. Okay? So these are some of the functions of all cells. Now this is very basic things. What is solution? Imagine this is a glass of water and we are pouring maybe sugar, salt, something. What would be in this the solution, the solute, and the solvent?
Solution is water and salt, and solute is salt. Solvent is water.
Okay, so this is the salt, and this is the water, and this.
Solution is the combination of water and the salt ocean. That one is sugar or salt. So one is the water.
Okay, so solution is the mixture, right, of two or more components. Okay, this is just so we don't get confused in the terms. Okay, solution is the mixture, as mentioned. The solute is the substance that is present in smaller amount. Okay, so the salt or the sugar. And the solvent is the dissolving medium, the water. Is the substance present in the largest amount in a solution. Okay? Why? So water is the body's main solvent. We mentioned 60% component in the cell is water. Now let's clarify some of the terms that I don't know, if you go to textbooks and you read a little bit, it could be a little bit sometimes confusing when we read about intracellular fluid and extracellular fluid. Okay, so what means intra? Anyone? Except you two. What is intra? Take a guess. Any guess is okay. One is intra and the other one is extra. Exactly the same. Intracellular fluid, extracellular fluid. Yeah, simple as that. There's no trick here, okay? Intra is inside, extra is outside. So, we mentioned in the first class, many of the medical terminology is composed of different parts. The parts will give you the idea what we're talking about, okay? Intra, inside, extra, outside. Cellular for cell, okay? This is just the fluid inside the cell or outside the cell. Okay, now that is clear, but where is inside the cell and where is outside the cell? Inside the cell, maybe it's not too difficult, right? Inside the cell is very straightforward, right? The nucleoplasm, and the cytoplasm, cytosol. But when we talk about the extracellular fluid, we can be referring to either the plasma. Here, this is a blood vessel. Imagine this is a blood vessel. The plasma, which is the liquid part of the blood. Okay, the blood has some cells, but also has some liquid part. This is the plasma. So that is extracellular fluid outside the cell. But another one is the interstitial fluid. Inter means between. Stitial is always referring to cells. So it's between the cells. Between the cells will be this space here. Okay? And this space also contains some substances like amino acids, vitamins, and so on. Could have hormones or neurotransmitters. Okay, this is just to make clear where is where. Okay? Whenever we mention. Maybe we don't have that much time. The video briefly mentioned also things about membrane transport. We have mentioned that the membrane is not solid, is not rigid. Either we imagine the membrane as containing the parts of the cell, things or molecules can go through the plasma membrane. Okay, but how do they move from the inside to the outside? So there are basically these two types of transport. Or one is the passive transport, and the other one is active. The only difference, the main difference between the passive and the active is if it's you need energy to transport. If you don't need energy, if it can be transported without energy, then it's passive. But if you need energy, meaning ATP, then it will be active. Okay? Basic, very rough difference. And we have different types of passive transport. This is a simple diffusion for certain elements, and facilitated diffusion. You can see here from some of the figures. We mentioned different concentrations. Molecules will move from areas where it's highly concentrated to less concentrated. That is the simple diffusion. Okay, they will go through the plasma membrane. If, of course, they are small enough to go through, if they can, if they are lipid soluble. So we have this simple diffusion. We also have facilitated diffusion when we are using some proteins that go from both ends of the plasma membrane. Sometimes it's not specific, meaning that if the particle, if the substance, if the molecule is small enough, or it's not charged or something, then they can go through. If we are talking about specific transportant, then they have to be a particular element that will be transported, such as glucose, for example. If we're talking about the movement of water, in that case, it's particularly called osmosis. Okay. Another type of passive transport is the one called filtration. We're not going into much of details for this type of transport. We'll probably see a little of this when we touch on the urinary system. But filtration is when water and solutes are forced through the membranes by hydrostatic pressure. This hydrostatic pressure comes mainly from the blood. So this takes place in the kidneys. Okay. We have another, here. So the molecule will move from the blood circulation into this other space. We don't need to know the details of this filtration. We will see more when we touch on
the sodium.
So we will see the next video, and we will end with this one. Okay, so pay attention again to the video.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel
that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and two of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay, so the main thing here when we talk about active transport is the last thing they say. It requires ATP, right? Uses ATP to carry the protein. And they're moving from low concentration to high concentration. That is where we need the ATP. Okay, so I think maybe we can stop here and we will continue. Okay. Let's check really quickly on the next two slides and then we will stop there. This is another way of active transport called exocytosis. And it's another way of moving substances outside the cell. This is an example of exocytosis and this is endocytosis. What would be the difference? Basically, you're moving substances either to the outside, that is exo means out, yes, or you're moving them inside, endocytosis, okay? So in the exocytosis, the product to be released, the substance that you are getting rid of, first will be packaged in a small membranous sac. So here is the substance. You surround it. And this vesicle, this is called the vesicle. And the vesicle will migrate, will move to the membrane, the outer membrane. Okay? It will fuse, meaning that these membranes will fuse with the plasma membrane and then will release the substance that it contains. This is how it will move substance. This is a method used by hormones, for example, when we release hormones or other cell products. And this is endocytosis. Remember, always endo will mean going inside. Okay? So you are basically taking something from the outside the cell, inside the cell. Different ways of doing it. Either phagocytosis, again, this is the plasma membrane. You will enclose the substance and create this vacuole and bring it inside the cell. Pinocytosis, when there are small substances, or it could be mediated by receptors, so very specific mechanism to engulf or to eat or bring inside these substances. Because it's very long and tedious, this next part, I will ask you to review by yourselves what is cell division. We will not cover it here, but please read about this. Mainly what is happening or what is going on, how they go from being only one cell to two cells. Okay, so the main phases and what's going on in each of the phases. Cell division is one, and the other one is the synthesis of proteins. How you go from having your DNA, remember we mentioned the DNA is the blueprint. So we have here all the instructions. how we go from the DNA to the protein, to having the protein. Cells have so many proteins that this is constantly happening in the cell. Okay, I think you can check this on any textbook. I would advise you maybe to have a figure or your own schematics. Okay, there are these number of phases. This is happening in one phase, another phase. Do your own drawings so that you can remember this. Okay, and we will start from here for next class. Okay, yes. If you want to rewatch the videos, remember again the links, the URLs are at the end of the handout. Okay? So only these two things, review by yourselves, protein synthesis and cell division. Okay? You may have studied this in Japanese, so probably just have to refresh your memory and put now the words in English. Okay? That's probably the heaviest workload that I'm going to give you in the semester, so please. Thank you very much. Any question? You want me to repeat something? The homework? Did you understand the homework? No? On the back? How about the back? Is it okay? You understand what you have to do? No? Yes?
One more.
Two things you have to do. Okay, two things. First, cell division. Okay, read about cell division. How many phases it has, what happens in the phases. Okay, so one is cell division. The second homework is protein synthesis. Okay, how do we go from the DNA to the protein? Remember? You don't have to memorize all the small details. I just want you to have the general idea. I have the DNA, will open, it will match with this, we will go this way, and so on. So if you figure out maybe this and understand this, it's probably enough. Yes? So two homeworks, cell division and protein. That's it. Okay? Any other questions? Is it okay?
All right.
Yeah. Anybody else? Are you okay? Yes? No? Okay. If no more questions, if you have other questions, send me email if you want to make sure. Okay? Otherwise,